So today I'm going to try and show you guys a video to try and capture this entire process of cellular respiration. Before we get going, you're going to need to gather some things. You're going to need 4 ADPs. You're going to need 4 inorganic phosphate molecules. You're going to need 2 NAD pluses, 2 hydrogen ions, and then you want to take 6 carbons and kind of link them together like this to form the 6-carbon compound glucose. Now glucose's formula is C6H12O6, but we're not going to worry about the oxygens and hydrogens. We're going to focus only on the carbon and what's happening with carbon, because remember, we get energy by breaking carbon-hydrogen bonds and forming carbon-oxygen bonds. And the first step is glycolysis. And so we're going to do that here at this lab table. And the gist of the process is shown here. We start with glucose, and we end with 2 molecules of pyruvate. And in the process, we get some ATP, and we get some NADH. Now, the question is, how does all that happen? Well, for that, we're going to flip to the next page of this packet, which you have. And you can see that glycolysis consists of 10 separate chemical reactions. The starting compound is a 6-carbon compound, glucose. And the end are 2 3-carbon compounds called pyruvate. The rest is really just detail, but there are a few things that I want to highlight. So first of all, you do need to invest 2 ATP at the start of this process. So we actually right away, we're at like -2 ATP because we're investing that, but we're going to pay those off later. And what happens is, right about here at reaction 4, this 6-carbon unit is broken into 2 3-carbon units. So I want to show you this over here. You can model this yourself. We start with glucose, 6 carbons, 1, 2, 3, 4, 5, 6. And at reaction 4, there's a reaction that kind of breaks these into 2 3-carbon units. So then if we go back to this page, we can see that a series of other reactions take place. We can see that NAD plus is put in to form NADH. We can see that ADP and phosphate combine to make ATP. And we see ADP and phosphate down here combined to make ATP. And so we can model that over here. We rearrange, break some bonds, form some new ones, rearrange some things, and essentially we get NAD plus swooping in and NADH coming out. We get an ADP and phosphate coming in, ATP coming out. We get an ADP and phosphate coming in, and ATP coming out. So far our net gain is we have 1 pyruvate molecule here, that 3-carbon compound. We've got 1 NADH, and we've got 2 ATP. But if you remember back here, this reaction, we actually broke the 6-carbon into 2 of these, which means everything from here on down happens twice. So we have to do all of that again for this second 3-carbon unit. NAD plus accepts an electron to become NADH. ADP and phosphate form ATP. ADP and phosphate form ATP. So by the time we're done, we have 2 3-carbon units of pyruvate, which we see on this diagram right here. We have 2 molecules of NADH, and it appears that there's 4 ATP, but remember, we had to invest 2, and so our net yield is 2 ATP, 2 NADH, and we have these pyruvate. And if we flip to the end of this packet, you can see that. We have 2 NADH and our 2 ATP. So after glycolysis is finished, we're going to move into the mitochondria. This butcher paper represents the mitochondria. Remember, the mitochondria has a double membrane. This outer membrane is called the outer membrane. Then we have the inner membrane, and the inner membrane has a series of integral membrane proteins embedded in there, including this very special one called ATP synthase that will come up later. All the interior space here is called the matrix, and we're going to set a few things up in here. First of all, In the matrix, you want this 4-carbon oxaloacetate. You want 1 FAD, 4 NAD pluses, 5 H pluses, an ADP, and an inorganic phosphate. Over here, you're going to want a little pile of beans. You know, nothing magical about the number at this point. And then the H plus ions, you want slightly more in the matrix than in the intermembrane space. So I have 4 hydrogen ions in the matrix and 2 in the intermembrane space. I have an oxygen atom molecule here in this box. And then I'm going to put an inorganic phosphate and an ADP inside ATP synthase like that to set us up for later. So the next phase is what's called the transition reaction. This is where pyruvate is turned into acetyl coenzyme A. You'll notice that a molecule of CO2 is released in this, and a molecule of NADH is produced. To model that, I'm going to take my pyruvate, which was 3 carbons long, and these 3 carbons are going to enter the mitochondria. And what happens in the transition reaction is essentially one of these carbons is broken off, and it's exhaled as CO2. So to get rid of that, we're just going to give a little chug. All right, that's gone. So now some energy has been released from the breaking of that bond, and we can couple that with the formation of NADH. So we have some more NADH from this process that's formed. If we go back to this page that shows our yields, it says 2 NADH, and you might wonder, well, why is it 2? And remember, there's 2 molecules of pyruvate formed from glycolysis, so this happens twice, once for each one. So that's how we get 2 NADH in this process. The next step is the Krebs cycle. Sometimes it's called the TCA, or tricarboxylic acid cycle. Whatever you call it, it's always represented as this like circular process. That's weird. We'll have to figure out why that is. In the TCA cycle, or the Krebs cycle, notice that 2 carbons are coming in as acetyl coenzyme A. 2 carbons are coming out as CO2. So we're continuing to break these bonds, and as we do, we can couple that with the production of some other molecules. We're going to get some ATP out of the deal. We're going to get some FADH2, and we're going to get some NADH. And if you flip to this page, we can find exactly where that's all happening. So backing up a little bit, here's our 3-carbon pyruvate from glycolysis. In the transition reaction, we broke one of those carbons off as CO2 to yield a 2-carbon acetyl-CoA, and we got some NADH out of that. And remember, all of this is happening times 2 because there's 2 molecules of pyruvate from glycolysis. What happens next is these 2 carbons of acetyl-CoA attach to this 4-carbon unit called oxaloacetate. So to represent that with our models, I'm just going to take these 2 carbons and just kind of attach them on. So now I've formed this 6-carbon unit, and this is in the mitochondrial matrix. And then if we see what happens, essentially we're going to carry out a series of chemical reactions. We're going to break some bonds, rearrange some molecules, and in the process we get 1, 2, 3 NADH formed, and we get 1 FADH2, and we get 1 ATP, and in the process we're basically breaking off 2 molecules of CO2. So to model that, we take this 6-carbon compound, and again we're going to break bonds, rearrange bonds, and in the process NAD plus will be reduced to NADH a second time and a third time. FAD will be reduced to FADH2, and in the process we're breaking off 2 molecules of carbon which are exhaled. That leaves us with this 4-carbon oxaloacetate, which is now ready to join 2 more carbons from the from glycolysis, and then this whole process happens a second time. So going back to this sheet, once again, this whole thing happens twice. So our total yield is actually 6 NADHs, 2 FADH2s, and 2 ATP. And if we flip here, we see that. 6 NADHs, 3 per round of the Krebs cycle, 2 FADH2s, 1 per round, 2 ATP, 1 per round. So at this point, the Krebs cycle is finished. All 6 carbons of glucose have been broken down and released as carbon dioxide, but the energy released from that breakdown has been coupled to the formation of a bunch of NADHs, and there's just an arbitrary number here. You can look at that green packet for the exact number. A little bit of FADH2s, and we got a couple ATPs out of the deal as well. But remember, the goal is ATP. That's the energy currency. So what we want to do now is take the energy in these molecules and convert that to ATP. So to do that, we need to use the electron transport chain. And if we come over here, the key to this whole thing is ATP synthase. ATP synthase has a binding site for ADP and phosphate, and theoretically, it would be great if it could just stick them together. But the problem is, this reaction has a positive delta G. It requires energy to do that, and right now there's no energy for this to do that. So what this system is all about, it's about trying to harness the energy in these reduced electron carriers to drive the production of ATP. So the way we're going to do it is we're going to take an NADH, and I'm going to use one of these beans as an electron. And what happens is NADH kind of interacts with this protein, and it passes its electron to this protein. The H plus is just kind of released into the matrix, and then this electron is going to be passed from protein to protein to protein to protein. That happens because each protein is slightly more electronegative than the last. Further, these blue ones are what are called proton pumps. So as that electron is passed, energy is going to be given off, and that's going to be used to transport a hydrogen ion against its concentration gradient. And I screwed that up already. So we're going to move some of these down here. There we go. So we have 3 up here and 4 down here. So against the concentration gradient. So what happens is, as this electron moves, this H plus is going to be transported across against its gradient. It's going to come to this protein. Another proton is going to be pumped against its concentration gradient. It's going to be passed to this protein, to this one, and pumped against its concentration gradient. Eventually, the electron is passed to oxygen, the final electron acceptor. Oxygen is going to combine with some hydrogen ions and that electron to form water. So now we have this huge concentration gradient. Unfortunately, there's no way for this charged H plus to get across the membrane. There's only one pathway through, and that pathway is through ATP synthase. So the concentration gradient is in this direction, so it travels down its concentration gradient, which is exergonic. That couples the endergonic combination of ADP and phosphate into ATP. Another ADP and another phosphate come in, another hydrogen ion, and another one. And this just keeps going and going and going, as long as NADHs and FADH2s are available to transport it. Now, one other thing I want to point out, each NADH will continue to dump its electrons here. FADH2, though, it doesn't fit in this protein. It's a little too wide. It doesn't interact with that one. It actually interacts with this one a little bit lower in the electron transport chain, and as a result, not as much energy is released by this, so FADH2 doesn't actually contribute the same number. So if we go back to our sheet, you can kind of see that process summarized here as NADH passes its electron and becomes NAD+, and the electron is transported down its gradient. You can see that we are pumping hydrogen ions, boom, 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 against their gradient, and then eventually they go that way through ATP synthase, where that energy is used to form ATP. And ultimately, each of these NADH2s that we produced end up giving us about 2.5 ATP for a total of 5 and 5. These 6 give us about 15. Our FADH2s give us a little bit less, about 1.5 each to 3, and that's how we get our totals calculated.